Good afternoon and welcome to NRGene's webinar series. My name is Yana and I'm happy to be your host. Today we are having a special webinar where, in addition to theoretical discussions and explanations, we'll actually see our CannaGENE in action. CannaGENE is a genomic database that reflects the cannabis complexity and diversity and allows improved planning of any research or breeding program. We are very excited to share it with you today. We will save time for questions at the end, and we encourage you to type in your question during the presentation using the Questions tab on your screen. This webinar is also recorded and will be posted on our website in case you want to listen to it once again. It's my pleasure to present today's speakers. I'd like to welcome Ronnie Rechtman, who is a senior bioinformatics scientist in the Genomic Analysis Services team of NRGene. She is responsible for several research projects in the field of comparative genomics, among them the development of NRGene's first version of CannaGENE that you are going to see today. Ronnie holds a master's degree in computational biology from the Weizmann Institute of Science and has an extensive experience with data science of human and plant genomics in the biotech industry. I'm very happy that she found time in her extremely busy schedule to join us today. Our next speaker is Dr. Uri Weishaus. Uri is with NRGene since 2011, applying genomic tools for marker development and breeding projects of various crops. In the last several years, he has specialized in cannabis genetics and became an expert in the field. Uri holds a PhD in plant and microbial genetics from the Hebrew University, and his academic background is in genetic of plant secondary metabolism pathways. Uri, the stage is yours.
Thank you, Yana, for the introduction. I'd like to start with a brief intro about NRGene. For those of you who are not familiar with what we do, so NRGene is not a cannabis company. We do not grow. We have no licenses to process cannabis as a plant. We take in DNA or data and tissue samples. And we usually work, most of our clients are in the seed industry, big seed companies. Most of the largest seed companies work with our tools, as well as other vertically integrated industries, usually relying on plant raw materials. Specifically in the cannabis and hemp space, we have close to 20 customers, most of the large LPs in Canada and several smaller companies in the US and in Canada, as well as a company in Switzerland and in Israel. So we can't do a webinar about cannabis without showing pretty pictures of this magnificent plant. So anybody who's grown more than a plant in his life knows that cannabis comes in all shapes and forms. The great diversity in this species is across plant morphology. You see all sorts of flowering types and all sorts of chemotypes. The plant is known for a huge range of secondary metabolites. So a lot of them are of interest to medical research and recreational use. But it doesn't end at the morphology or chemotype. Obviously, there's a genetic background to this. So there have been several attempts to characterize groups and to sort the great diversity of this plant species into groups. And in this example here has been, this is published from 2016 by Weiblin's group, separating the different types of plant morphologies or characteristics into certain groups, and hemp, which is a type of cannabis, fits in one of the groups. And if you go into CBD hemp, you're kind of going on the border between multiple groups. But not only is this plant very, very hyper diverse on the genetic level, it's also highly heterozygous, mainly because it's being propagated as clones, but also because of the dioecious nature of male and female plants. The plant prefers to be outcrossed. So most of the analysis done so far have been on plants that are almost fully heterozygous, or at least mostly heterozygous. So why did we even set up CannaGENE? The basis of analyzing genetic information in cannabis and in any other crop for that matter is data organization and standardization. The basic need for any crop to be able to do genetic analysis and research is a good reference genome, the more the merrier, and then comparison of multiple genomes, and exploration of diversity usually at lower resolution, and then ability to detect genotyping. And on top of that, you need ability to do prediction, and all of that is bundled into what we call molecular breeding. So the first need is data organization and standardization. Then you need more data, which is de novo assemblies of reference level genomes. And we need to compare coherence between genomes and organize the data in a uniform structure. And obviously enable accurate gene level research, because most of the breeding and applications of genomics is at the gene level. So talking about public reference genomes, we could see that there have been at least 4 publicly available chromosome level cannabis genomes released to the public domain. And there's a great difference in the overall size of it ranges from about 600 megabase pairs to closer to 1 gigabase pair. And the difference between the 4 assemblies is partly due to biology. But I would have to guess, and there's a big gap in time between the release of all these genomes, also technique and assembly quality of putting all the pieces together into the complicated puzzle that we call a genome assembly. So the bigger fraction of the unmapped chromosome regions, meaning reads, short reads that were not mapped to any of the chromosome, pseudochromosome assemblies, means that the assembly was not as successful. So there's a big difference between the 4 cannabis genomes. So NRGene addresses that by using our own proprietary method of assembling good reference genome. So it starts with a unique cocktail of up to 8 types of libraries that we use, most of them on the Illumina platform, and recently we've also added PacBio long reads. We overall make a 210x coverage of the genome, illustrated in this schematic here. So it covers in great redundancy all of the genome. The main advantages of using this approach is that it enables us to resolve highly complex, highly repetitive regions in great amount of success. It also allows us to phase the 2 haplotypes in the genome, and I'll get into that in a second. So when I say a phased genome, I mentioned earlier that most of what is analyzed is a heterozygous material. So a heterozygous material usually has most of the genome with 2 copies, maternal and paternal, illustrated here with these little dots or circles. So if there's a difference between what comes from the pollen donor and the female parent, a traditional short read assembly would not phase it, will create a collapsed assembly, taking either both in bits of information, combining them into a false sequence, or taking half of the information from one of the parents or the other, and thereby losing half the information. A phased genome assembly takes into account both parental haplotypes and separates them. So this is how NRGene creates its phase assemblies, and we are showing here statistics of both the unphased and the phased assemblies of same genomes. So these are the range of results that we typically get for cannabis and hemp genomes. And what I would like to point your attention to is the Busco score. And Busco is a measurement of the completeness of assembly, or the measurement of completeness of a set of known genes that are well conserved in the plant world in this case. So as you can see, the scores are pretty high, 95 to 97% of complete Busco genes, and then multiple copies of the same Busco genes could also be detected in the genome. But if you look at the phased genomes, the same genome, the numbers go up, meaning we have been able to detect more versions of the genes in the Busco set, and also there's multiple copies of single and multiple duplicated genomes fraction. So the phasing allows to detect more versions of the same gene content. Another thing we addressed when build up the database is the arrangement of chromosomes and nomenclature. There's a need to do uniform mapping between chromosomes. So we took CBDRX, Sunrise Genetics CS10 genome, and we did a lot of work on improving it. There's been previous versions released, V4 for Purple Kush, V2 for Finola, and CBDRX had recently released last April a version that changed altogether the nomenclature, and it is very likely to be accepted as the gold standard of cannabis nomenclature. They arranged it by size. So 1 became X and so on, the sex chromosome. And there's also an update for Purple Kush version 5, which is constantly, so the versions for the chromosome naming have changed over the last 3 years in the public available assemblies. So there's also a need to fix the coordinate system on one set of coordinates and one fixed nomenclature for chromosome numbers. So we did that in the CannaGENE database. Another thing we did is improve the contiguity of the scaffolds. So this is a comparison of chromosome 6, a classic before and after picture where we compared elite scaffolds. So NRGene Elite 2 is one of our genome assemblies that NRGene has assembled of proprietary genome. And we've compared that to the scaffolds of chromosome 6 in this region in CBDRX CS10. So you have the two phases of the blue and the orange phase, A and phase B. And this is done in several ways. One of them is gene synteny. So each dot here is a gene. So as you can see in the before and in after, doing this type of comparison and crossing that with two other methods allowed us to get better contiguity in both phases and get one continuous scaffold for the NRGene Elite 2 and Elite 1 assemblies. So the next step we did is we know that there is great differences in transcripts, gene content, and transcript level. So the more transcript data we could combine into the database, the more information we could look at and compare. So we took 4 data sets from publicly available transcript data sets, some of them from the CBDRX, also curated by proteomics. And we've made a non-redundant transcript data set. And once we had that, we could align each of that and make an annotation to each of the genomes in the database. So what is CannaGENE? Basically, it is a vast curated cannabis database which contains unique proprietary genomes, NRGene 1 and 2, and along with 8 other genomes and 4 publicly available genomes. And pretty soon in the first update of this database, we will upload 42 additional genomes into the system. So with this type of data organization, you could take a region of interest that you're interested in from literature or from QTL mapping or any other indication that this is an important region, and zoom into the gene level at the highest resolution and accuracy possible and look at regulatory elements, present absence variations, copy number variation, and novel alleles and novel genes that are present in our data set. So we have arranged, and we're using the IGV by the Broad Institute as the visualization tool, and we've modified it to present multiple tabs and allow for the comparison and visualization on one set of coordinates, and this here CS10 reordered, everything in the database. The 4 public reference genomes, the 2 phase genomes from NRGene, and the 8 whole genome shotgun sequencing of 8 genomes. And we've also done a track of variant calls for each line in the database, compared them to the 4 public genomes, added an annotation data track, and also a track of candidate genes from the literature and other sources that are interested to cannabis research. So CannaGENE not only arranges the data and organizes the data in a very high utility way, there's also 50 hours of bioinformatics support and consultation. So we set it up for you on a local system, and we help teach, train using the system, and also there are 50 hours that you could use for whatever bioinformatic needs you have. So you could request specific tasks from our professional team as part of the service hours. And we commit to 2 database updates per year that add in more data and more features as we develop. So now I'll give the stage to Ronnie, who will show you a live demo of our CannaGENE.
Thank you, Uri. I'm going to present CannaGENE online and show you how to access the data for visualization and analysis. We selected IGV tool from the Broad Institute as the gateway and viewer for CannaGENE data. We in NRGene modified it in order to support multi-genome comparison and develop some other extension to this tool. What you see here is an IGV session. This view is based on this CS10 reordered coordinate system as the reference genome, and we see here chromosome 10 in specific location. In this view, there are several tracks of data and information loaded and mapped to this specific location. And here you see a place for attributes. Here it's possible to load phenotypes like flowering time and other traits of the samples. Since they are not relevant to this example, I'm going to hide it. And you see here many tabs, multiple tabs. Now we are in the tab of NRGene, one of NRGene's proprietary assembly. It is called NRGene Elite 1. And all the tracks that we uploaded to this tab are relevant to NRGene Elite 1 data, sequence, and analysis results. I'm going to go over some tracks. Let's start with the sequence. This is the actual DNA sequence of the reference genome. If we zoom in, we can see the actual nucleotide sequence. And it's also possible to see the translation into amino acids and see different reading frames, possible reading frames of the protein. And it is also possible to flip the strand and see the sequence of the reverse strand. The first track here in the bottom is an annotation track. We took the Cannabis transcript database and tried to map it to the reference genome. So each one of the transcripts in the database is mapped to the genome and presented here with all the data and analysis results behind the pipeline. For example, the homology of the original mRNA transcript to the genome. This is the public identifier of the transcript, XM, from RefSec from GenBank. Also, we have the link to the original external NCBI resource. And we took the functional description of this gene from the public database. This is a calcium-dependent mitochondrial protein. And also the cluster members, the homolog, the paralogs, the other transcript in our database that were found similar to this transcript. So the data is here. And in this track, if we zoom out, we see many other transcripts that were mapped as annotation to the genome, where the color signifies the level of mRNA sequence identity. Now we will focus on the Contigs track. So here you see 2 Contigs aligned to the same location on the reference genome, which makes a lot of sense because the assembly is phased and contains both alleles of the line. If we zoom in a bit more, we can see in gray color the sequence which is identical to the reference, and in these colors are places where the sequence varies from the reference. For example, here we see an A that was replaced by C. But you can see that it happened only in one of the alleles. This one has the variation, possibly mutation, and this one has the sequence which is identical to the reference. Which leads us to the last track, which is the variant recalling track. It shows the result of a complex pipeline of variant recalling, which is based on a lot of data, including the raw read sequencing data. And the output is recalling a prediction of a variant. In this place, we have a heterozygous variant, which is colored in red. And here, in dark blue, we see a place that we detected a variant in one of the other samples of CannaGENEs, but here in NRGene Elite 1, the sequence is identical to the reference on both alleles. So to summarize, what you see here is two different haplotypes, two alleles. One is identical to the reference and one is different from the reference, having many mutations. All of them are on this location of the gene that we identified in this region. Now we can see the same location, the exact same location. If I go to another tab, for example, this is NRGene WGS1, another proprietary assembly of NRGene with different contigs, much smaller. So we go to here, we get the same location, and you see again the annotation, the alignment of contigs with slightly different haplotypes, and the track of variant calling. Here you see also light blue color for homozygous mutations, like this deletion here that occurred in both alleles as compared to the reference. So these three nucleotides or two nucleotides were deleted in both alleles.
So first use case example is a gene called GIGANTEA in Arabidopsis. So in Arabidopsis, it creates a fantastic phenotype. It is one of the key controllers, so this is GI, GIGANTEA, master controller of the transition between the vegetative and flowering state. So we thought this would be a good start to look at literature genes that control flowering time. What do they look like, or what do their homologs look like in cannabis? The phenotype of GIGANTEA mutation is a monster gigantic flower because it's stuck in vegetative state. It does not flower, and therefore the leaves keep on growing. So there's a homolog of this gene in cannabis, and Ronnie will show us what it looks like in the real demo.
Now let's see how we visualize the different version of GIGANTEA gene and different germplasm in CannaGENE. I'll use the text search. I will type the gene name, GIGANTEA, in this text box and jump to NRGene Elite 1 tab. Here I get the coordinates of the annotation of GIGANTEA genes in chromosome 1, this location. And you can see that in addition to the cannabis transcript database track, we have also a candidate gene track. Here we put some specifically selected, very interesting genes in cannabis related to flowering time or cannabinoid pathways, or things that may be interesting for research. So this gene appears here, the actual transcript from the database with its description. And you see that for NRGene Elite 1, we see this gray color here. It is part of a very long contig with no difference at all from the reference sequence. So this gene is super conserved. You see here, everything is blue color, meaning all variants are having the same genotype as the reference, and also in the aligned contig. Now let's check the comparative tab. Here I put together all the variant calling tracks from all germplasms in CannaGENE, all the different tabs. And you see that there are tracks which are blue, and also some with a lot of red variants, which are heterozygote variants, and also homozygote variants. I can select one of these germplasms, for example, WGS5, which is blue in most of its variant, but let's go have a closer look. This is the gene. This is the contig of NRGene WGS5. You see that it is very conserved, aligned almost perfectly to the reference, but there is a specific variant here in light blue. Let's zoom in a little. Okay, so we see here two variants, two possible mutations. One is a SNP. This is an A, which was replaced by C. And this is a sign for insertion. We can see in this location an insertion of 3 nucleotides that were inserted as compared to the reference sequence. These two suggested mutations, of course, they occur in an exon of a very conserved gene. So they suggest possible functional implication, which can be further researched in the lab for validation or further research in order to understand the implication of these mutations.
Thank you, Roni. And now we'll show another example of CannaGENE specific use case gene. This is OLS, olivetolic acid synthase. This gene is actually the metabolite gatekeeper of the cannabinoid pathway. It exists in multiple copies. I think the most we've seen is 9 copies in one of the genomes, in the cannabis genomes, and is responsible for manufacturing olivetolic acid. So any mutations, different variations, more effective versions of this enzyme may lead to a bigger flux of carbon products into the cannabinoid pathway. And therefore it has huge implications on, or may have huge implications on yield improvement and yield related traits going for cannabinoid production. So I'll bring the broadcast back to Roni. Roni, please.
We see here 3 copies of OLS gene. You see here 1, 2, 3. All are on the candidate gene track. So the same transcript, same original query transcript was mapped 3 times to the reference genome. And even here, you can see that it looks like tandem duplication or maybe inversion or something like this. In addition to the candidate gene track, we see also the Cannabis transcript DB, the full database of transcript. So you see that there are several versions from different germplasm and different sources to the same gene. So let's zoom in to the first copy with the highest mRNA homology. And you can clearly see that this transcript from Finola has a longer version than the original candidate gene. We can zoom in even more to the region of the end of the gene. And let's zoom in and see what you see here are alignments from different germplasm that we collected together and moved to the same tab in order to do the comparison. And there is an insertion right here and heterozygous insertion in NRGene Elite 1 Here, the insertion is a heterozygote in NRGene Elite 2, which is another phased assembly of NRGene. And these contigs are from the public assemblies of Finola and Purple Kush. And we see that indeed this region of the insertion, we see here stop codon and even 2 consecutive stop codons. So these stop codons in the reading frame may be alternative and create 2 different versions of these genes in different lengths. So again, this is another example to a case that we can further research, explore, take to the lab, and understand what's going on and whether it has any functional implication and whether it can be validated.
So we usually keep all our questions to the end, but there is one that's really relevant to what Ronnie has showed us. And the question is, why do you see multiple contigs in some lines and only 1 contig in others? Can you explain this to us, Ronnie?
Yeah, sure. Thank you for the question. The places here where you see 2 contigs, these are of NRGene's proprietary phased assemblies. They are phased and they contain both alleles. This is why you see 2 contigs. And here in Finola and Purple Kush, the assemblies are collapsed. So there is only a single contig for every genomic region. And this is actually one of the big advantages of NRGene's assemblies and data, that it enables the researcher to see the different phases and to analyze the variants of the different alleles. Thank you.
Another application that we have on the CannaGENE database is for synthetic biology. So when you look at synthetic biology, it's no longer very efficient to look manually over case by case on each copy of the gene. For example, the cannabinoid synthesis gene, biosynthesis genes, CBD synthase or THC synthase, they're THC-AS, CBD-AS. They're very similar genes with CBC-AS in the middle. There are multiple copies of functional and non-functional genes in each of the genomes in our database. So it's not very easy to look at it on a zoom in feature. It's much more efficient to have command lines and scripts analyze bulks of data. So we could do that as part of the CannaGENE package. We create analysis like this. We identify all copies of homologous genes that look like, and you determine the level of similarity to THC-AS gene. And in this case, we have done this for THC-A synthase, and we have over 17 new versions of this, or homologs to this gene, that clade into different clades. They're all unique versions that come from our database, and they are similar to CBD synthase, but they do other things we don't know. So what they do, we don't do biochemistry in our wet lab, but these are accessible in the database and could be explored when using the system. Another example in CannaGENE is phytochrome A. We've looked at genes from the literature that control flowering time. Specifically, phytochrome A has been known to be the far red sensor in Arabidopsis and many other plants. It senses the days getting shorter and triggers a cascade of signals for flowering. So we looked at what's the status of this transcript, or the expression level or expression presence of this gene in the various lines in our database, and we saw that all of the lines have functional copies, highly super conserved, 100% on the protein level and almost 100% on the DNA level, except one of the lines in our database, which is Finola. It does not have a functional copy of the gene, and it is an autoflowering variety. So is this the autoflower gene? Maybe. So this is an example of how you could look at the literature, explore the database, and then go back to the lab or the field and start doing test crosses and biochemical research and continue in silico research to get real inside information and ask biological questions. So the main uses of CannaGENE, I think, is for early stage research. Companies and institutions with or without cultivation license that are maybe just beginning to explore their cannabis database, or they explore their cannabis germplasm or have no germplasm yet. If you're also missing qualified personnel to deal with genomics data, you don't have the infrastructure or the relevant people, CannaGENE can take care of that as well. It's a complete package, so you could use the consultation hours to close this gap. And then if you do have these things, cultivation licenses, all the germplasm relevant and personnel people to explore, you could also use the data to integrate personal data and private proprietary data in a private version of the database. So you could compare and analyze in the same way and efficiency your own personal data with information in our database and the good arrangement that makes it easy to drill down at to the gene level. The main applications that people have asked about CannaGENE or used CannaGENE for are, first of all, synthetic biology, mining of the database for novel alleles or novel genes for discovery and cloning. People in this space are not very interested in the plant. They just want new sequences or different versions of the enzymes to clone. So this is the ideal tool to do so. Breeding, if you could combine regions of interest, marker, convert genes into fast genetic markers for breeding, or combine it with QTL data, either done with us with our quick genetics package or done otherwise and made more efficient by looking at multiple varieties. And then there's gene editing. A lot of companies in the cannabis space are interested in genome editing. So if you're doing genome editing, you need to identify your targets, you need to identify your off targets, and you need to design your guide RNA. And once the process is done, you also need to validate the edit. So for all of that, you need high resolution, gene level, accurate gene content information. So all of that is provided by CannaGENE. So to sum up, a lot of companies or academic researchers can benefit from having CannaGENE. There's a recap here on this slide of all the uses and advantages of using CannaGENE. I'm going to leave this slide in the background, and with that, we'll be happy to take questions.
We do have a few questions, and the first one is, if I have my own personal data, can it be integrated into your database?
So yes, you could do one of two things. One is have the regular database, and with a minor additional cost, we could integrate all sorts of data onto the database. And we do that for a client, so it remains with the client. The other version is originally make an entire database with your database integrated, your personal data integrated into the database, and becomes a private version belonging only to the client.
Another question is, does NRGene provide custom data generation, for example, RNA-seq?
Yes, so this is also relating to the first question, where we don't produce RNA-seq ourselves, but we have a bunch of providers that we work with and have been validated by us to provide all sorts of transcript data, RNA-seq, proteomics, anything that is in good standards of quality of data, we could consider integrating, and most of the things we could integrate into a private version of the database.
Uri, our attendees want to know, what does the consultation hours of bioinformatician include?
Okay, so the consultation hours include projects that are within the scope of our bioinformatics team. For example, looking at the literature and finding homologs in cannabis or in the multiple lines of cannabis and getting a report with all the coordinates in their system and the sequences, things. If it's within the reasonable parameters of bioinformatics challenges that the technology exists, we can do it and provide answers.
And then it probably will be the last question: who owns the IP generated through the database?
Okay, so this is an easy one. When you license CannaGENE, you get the data, and anything you find belongs to the client. You own everything that you find.
I'm sorry we couldn't answer all your questions, but please know that we will follow up by email to answer all of them after the webinar. I'd like to thank Ronnie and Uri for their time today and the terrific presentation they shared. This webinar was recorded and will have it available in the next few days, so keep an eye for our email with a link to the recording. You'll be also able to find it on our website. If you have further questions or an interesting research project and like to discuss it with our experts, please don't hesitate to contact us at nrgene.com. Thank you for joining us today and enjoy the rest of your day.